biology, so it's nice to see you guys. There's also a few new faces that I've actually seen around campus and probably waved to, but hi, I'm Steve and I teach into the molecular biology, the genetics side of things. So today I'm going to take a little bit of a spin on what Jane has already given you, and I know she's given you information about these particular hemoglobin opathies and thalassemia and a whole range of different things. But today I'm going to actually look at the genetics and the molecular side of it and how you would diagnose or detect these specific blood diseases. And it's really important that we look at, and I know I've said this in class many times, it's really important to understand the genetics in terms of its structure and function to be able to understand how you would first of all design a diagnostic test to test for a specific disease. Then how would you analyse that? If you don't understand the basic structure and function of a gene and how it works at a molecular level, it's really difficult to do that. So what I thought I would do is just take you on a little kind of journey, and I know that some of you, my students that I already teach, have been on this journey before, but I've kind of added some new things for you guys anyway. Just to give you an idea on why it is important to understand these things first. So what we're going to talk about today is really to understand how loss of function and gain of function contributes to these particular diseases. Important to understand why gain of function works and why loss of function works in these particular disease states at that structural level of the gene. Appreciate that mutations affect any step in the pathway of globin gene expression. So these mutations don't necessarily hit one spot. They affect a whole range of different components of the gene, whether it's in a promoter region, a regulatory region, enhancer region upstream, whether it's in splice junctions, where the intron-exon splice junctions, whether it's in coding regions, whether it's in tartar boxes, whether it's in transcription initiation start sites, that whole range of different things. All of these mutations can affect the level of expression of a particular gene. And also we're going to appreciate the many forms of molecular techniques used to detect and diagnose these particular diseases. Now, some of these you'll look at and you'll go, wow, what is that? You know, it's quite complex. But I just want to give you a flavour. I don't want you to go into any deep detail on these techniques. We'll actually be doing one of these techniques in human molecular biology towards the end of the semester, which I'll allude to in the talk. But just get a general idea. I know you're doing gel electrophoresis, so they're probably the ones you want to concentrate on. In terms of the other ones, just have a general look at them. So today we're going to discuss the thalassemia, alpha and beta, and the hemoglobinopathies, sickle cell disease and some others, and the molecular and cytogenetic analysis and diagnostics of those particular diseases. So the first thing I want to really talk to you about is this whole concept of loss of function and gain of function. And this is what occurs in these diseases that Jane's been talking about. We either have a loss of function or we have a gain of function. And the most common effect is loss of function. Now, if you look here on the left, you'll have two alleles, and they're both producing, they both have a gene that's producing a particular protein that has a function. And you can see this by all of these little dots in here. This is the level of function that that particular protein has. Now, if you're a heterozygote, so if you have a mutation in one of the alleles, you produce less of that particular protein. Whereas the other allele, you're producing the same amount. And in many disease types, that's enough. There's a compensatory effect. You're actually getting enough production here of protein to be okay. However, if you're homozygous and you lose the effect of both of these alleles, you produce a lot less of that particular protein. So you lose the function. You can lose the function altogether. Or you can just lose the amount of protein that's actually made for a particular process. If you're looking at gain of function, it's the opposite. So again, you have your two alleles producing a certain amount of protein. You have a mutation in one allele, and it produces a protein that has a different function. It's the same allele, but there's a mutation there that produces a very, very similar protein, but now that protein has a different function from what it had before. Again, if you have both alleles are mutated, you're producing a lot more of that particular protein. So gain of function is not necessarily a good thing because you're gaining a function from a protein that had a specific role in a cell. That specific protein has now gained a new function, which could be deleterious to the cell or it could be beneficial. And it's the whole, looking at the whole evolutionary process, natural selection, that's how that occurs. Now, one of the classic loss of functions is thalassemia, and it's a loss of function due to the deletion and subsequent reduction in gene dosage in alpha thalassemia. And it's commonly due to the deletion of the alpha globulin genes. So what I mean by gene dosage, what I mean is that dosage and that protein production is less. So that dosage is less. So you're getting less dosage of that particular protein, which can be which in this case is deleterious to the cell. A complete loss of function is exemplified by beta thalassemia, and that is a complete loss of function. There's not usually any gene dosage effect. It's gone. That function is completely gone. And the severity of the disease is due to these particular loss of function. So if you've got a slight loss of function, you may have a severity that is not as severe as a complete loss of function, obviously, and so on. So it's all about gene dosage. And there's a whole range of diseases, as you're probably aware, that fall under this. So gain of function mutations, again, more is not better, as I said. These are mutations that enhance one or more of the normal functions of a protein. So it enhances its function, makes it work even better in a cellular context, but not so much for the cell because the cell doesn't like it. It could be deleterious to the cell, or it gives it a completely new function. One of the hemoglobin Kempsey is a good example of that, where it results in a mutation in the hemoglobin structure that binds oxygen with such a high affinity. And you probably remember from your early studies with hemoglobin that oxygen affinity is low for the hemoglobin molecule, and then gradually it's a cooperative effect where you get one molecule of oxygen binding, you get conformational change in the hemoglobin, another one binds, and then another one binds, and finally you get this really nice affinity of oxygen for hemoglobin. However, in hemoglobin Kempsey, this just locks it into that high affinity state all the time. So you can't deliver any oxygen to your tissues. So that kind of has given you a bit of an example of loss of function and gain of function. Have any of you guys here heard of this concept before? Loss of function, gain of function? No? Okay. So it puts it into more context, right? Yep. Okay, so I know that Jane has given you, and I'm not going to go through all this stuff about the structure of hemoglobin because Jane's given you perfect examples on all that. But it's a basically 4 peptides, 2 alpha and 2 beta peptides that form this particular structure that is essential for carrying oxygen. Now, another thing to really understanding why mutations can cause disease is understanding conservation of proteins across species and across evolution. And this is one thing that's really important to understand about this whole concept of selective pressure. If you look here, you've got primates and so on. You've got a whole range of different things, dogs, goats, elephants, and this is the hemoglobin molecule, and you can see how highly conserved it is across all of those species. So there's a strong selection pressure in nature for it to maintain that particular structure because it is such an important molecule, right? So you can imagine that any mutation is going to be problematic. So trying to understand mutations at just a gene level is difficult enough. But putting it into a context on looking at how proteins are conserved across species in evolution gives you an idea of the selective pressure on that particular protein to maintain its function. It doesn't want to change. It already does what it does very well. So any mutation is going to cause problems, as you can imagine. Now, you probably have seen something similar to this, that we have a whole range of flavours of globin genes, and if you look here, we've got embryonic, fetal, adult, we've got some unknown genes with some unknown function, and we've got pseudogenes as well, which we talked about in first lecture for human molecular biology. But there's a whole range of different globins that are expressed during various stages of development, and I'm sure that Jane's probably gone through that with you. But it's important to understand this as well in terms of where these mutations are in these particular genes. And this is the whole concept of what's called global switching. Now, if you have the post-conceptual age to birth and then the postnatal age, you see that we express these globins at different levels. Some are switched on, some are switched off. For example, we have the alpha and the gamma, which is the fetal hemoglobin, which is highly expressed in the post-conceptual age. But as we're born and we develop, this expression of the fetal hemoglobin goes down and our beta globin goes up into the adult form, which we know as alpha and beta forms. Okay, so this global switching is quite important because this is where Jane alluded to earlier about the whole therapy, the genetic therapy, and looking at switching back on this particular fetal globin gene later on in life for people that are missing, for example, one of their, the beta globulin gene. Being able to genetically engineer and switch that back on. And there's research that's going on doing that now. Okay, is everyone with me? No problems? So I want to just talk to you about, I kind of at the start of the lecture I said that, you know, these mutations can affect any part of the gene. A whole range of different functions, the regulatory regions, promoter regions, exonic regions. But if you look at the beta globin gene, it's controlled by what's called this locus control region. So this locus control region is really hot topic at the moment for diagnosing and detecting some of these mutants, but also as a genetic therapy. So this locus control region has this DNase 1 sensitive site, which basically configures this in an open chromatin state. And you guys remember what happens when chromatin is open versus closed. So when your chromatin is open, you allow various transcription factors and things to come in and bind and regulate gene expression. When chromatin is tightly packed, it's silenced. So this is a really important region. And there is a Hispanic epsilon gamma delta beta thalassemia where this whole region is deleted. So there's no expression at all of this beta globin gene because it's locked in that tight state where nothing can get in. So you can imagine if you can reopen this by introducing regions in here, in there to make it sensitive to opening, then you may be able to switch this back on. It's there. The gene is there. It's just not working. So that's an area that is really interesting at the moment. So the clinical significance of this I just probably went into, but it's really threefold. Patients with deletions in this locus control region. So do everyone know what a locus is? Do you want to have a go? A locus? My students will know it, but they're not saying anything, right? No. Okay, so your locus control region, a locus on a gene is a particular region where it has a number of genes that are sometimes in families related to each other, sometimes not, but it's a region on a chromosome that contains a bunch of genes that are involved in a bunch of processes, and they're always controlled at this particular region. So patients with deletions in the locus control region fail to express the genes of the beta globin cluster. They're there, the gene's there, They just don't have this region. So can we switch that region back on? Can we introduce sensitive sites to reopen that chromatin to allow it to be switched back on? Yes, we can. We can do that in the lab. Components of the locus control region are likely to be essential in gene therapy, as I just said, and you can understand. And knowledge of the molecular mechanisms that underlie this globin switching. Remember, we just talked about this globin switching from fetal to adult globin. Makes it feasible to try and somehow manipulate this and upregulate it, to upregulate the expression of the gamma globin gene in patients with beta thalassemia, because it's been shown that alpha and gamma is an effective oxygen carrier. So if we can somehow introduce those locus control regions into the fetal globin, which is not expressed anymore, the gene's still there, it's just not switched on anymore, we can perhaps switch that on. Or we can perhaps just switch the beta one back on. So that's some of the research that's going on at the moment. So, everyone with me? Good? Exciting stuff, right? Yeah? That's good. Okay, so we're going to talk a little bit now about hereditary disorders. They're really divided into three groups, and I'm not sure if Jane has gone through these, but just briefly, there's the structural variants, which alter amino acid properties of the particular globin genes. So there's also thalassemias, and we talked about those. Thalassemias in terms of decreasing the abundance of one or more of the globin chains. So you can have mutations in a whole range of areas, promoter mutations. Do you remember what promoters are? You guys know what promoters are? What about some of the newer people that I don't quite know? I'm not going to pick on anyone, but does anyone know what promoters are? Yep, they drive gene expression. Really important in driving gene expression, right? So, many of the mutations that actually account for some of these thalassemias occur in mutations in promoter regions, in regulatory regions that switch on and off the genes. And also hereditary persistence of this fetal hemoglobin and conditions that impair basically that switch from fetal globin to the beta globin or the adult form. So there's the kind of three broad groups. And I think you've probably already seen this. There's many variants resulting in point mutations. We know what point mutations are mutations that change one nucleotide at one point in the gene. And you know about what happens. You can change a point mutation in the DNA and it can have no effect, right? Why might that be?
Same time.
Sorry?
Chain of codon in the same, the codon that produce the same amino acid.
Yep, perfect. Because of the redundancy in the genetic code, right? Fantastic. So we can have, but if a point mutation occurs in a codon region that changes that codon, then we're in trouble. It can introduce early stop codons, it can introduce a new amino acid, that a new amino acid will have different properties from the previous amino acid, it can change the folding structure, the whole conformation of the protein. More than 400 abnormal hemoglobins are described, 50% which are clinical, and they're really separated into three classes: variants that cause hemolytic anemia, so the bursting of your blood cells, altered oxygen transport, which we just saw earlier with the hemoglobin Kempsey, and variants due to mutations in the coding region that cause reduced or less affected globin synthesis. And here are some of the examples, and of course, I'm sorry you can't read that and you're not expected to know all this, but it just gives you an idea of some of the major classes of hemoglobin structural variants. Hemoglobin S, single nucleotide on the beta chain, and what its pathophysiological effects are, and so on. So you can have a look at those in your own time. There's a whole range, and on Jane's last slide, you saw all of those particular different forms or flavours. There's so many of them. Now, you've probably, sickle cell's always used because it's a nice, easy one to look at. And it's really well established. We know what the issue is. But if you look at sickle cell hemoglobin anemia, sickle cell disease, It's basically here where you have normally a T to an A, which is a glutamic acid, and you get a change from an A to T, where you'll have a mutant, so you'll get this valine. So you're going from a glutamic acid, which is a charged molecule, to a valine, which is a neutral molecule. So that's going to affect how this folds, and it does. And the reason why I'm showing this is because there's a way to detect this later on with restriction length polymorphisms. I know it's a mouthful, but I'll show you how that works. It's not too difficult. But we can detect this change molecularly in the lab, okay? And also, the alpha thalassemias, there's some really common things that happen with these guys. Most common form of alpha thalassemia is a result of gene deletions on the chromosome 16. So you'll have the gene here, and it'll be deleted where you've only just got now this guy here, and these two are deleted. Or there's this gene triplex complex as well, where you get an increased copy number of the particular genes, which is quite rare. And again, some beta thalassemias, mostly due to single base substitutions that you can have a look at in your own time. Okay? And for the beta thalassemias, there's more than 200 mutations described. Now, as I said earlier, these mutations affect a whole range of things in the gene. Transcription. So they'll affect the transcription initiation start site. So the transcription initiation start site, really important. DNA polymerase moves along, bang, hits the transcription initiation start site, starts translating out messenger RNA. It's a set sequence that is recognised by the DNA polymerase. So it's important that if this transcription initiation start site is mutated in any way, you're not going to get transcription of that particular message, and you're not going to make the protein. RNA splicing, we talked about that just earlier. RNA splicing, so splicing out introns. So there's specific nucleotides that you probably already know about that are GC and AT that are really important as markers for splicing out introns. So if they're mutated, you don't get splicing of introns. The introns stay intact, changes the whole reading frame, right? There's also capside. You guys remember what the capside is? Yeah? You're shaking your head? That's great. So the capside's really important on the message. The messenger RNA molecule has that cap, which is really important in directing it to the ribosomes, but really important also to protect it from degradation. Mutations in that capside can cause a whole range of things. RNA cleavage, I'm not going to go through all these, frame shift, there's a whole range of things. But as you can see, all of these spots are where you can get potential mutations and where mutations have been found that affect the transcription and translation of these particular disorders. So it's really complex. And you can imagine the tests and the diagnostic tests to try and identify all of these is really difficult. That's why there's not a lot of tests for all of these kinds, because they're really difficult to design, because there's just so many mutations. So more tests are designed for most of the common kinds of things, sickle cell anemia and so forth. More of the rarer ones, not so much. Really hard to try and figure out how you would detect a really rare form of the particular disease. And that's where it's important to understand genetics, gene structure and function, so you can work on how to do that. Also, and I'm not going to go through this, there's a whole range of splicing. We just talked about that. I don't really want to harp on that, but there's a whole range of splicing mutations that occur, again, that cause these issues. So now I'm going to get to, so now I've just gave you a kind of a brief genetics-based molecular sort of idea on structure and function, why it's so important, loss of function, gain of function, and why it's so important in these particular diseases, where there's mutations. Now I want to go through some of the techniques that are used to identify them, okay? So we're going to talk today about some protein chemistry, and these HPLC, IEF and so forth will become more evident in a minute, and we're going to look at some molecular techniques, such as the new droplet digital PCR and a whole range of things. And I know you haven't probably done a lot of PCR, so some of this is going to be a bit staggering, but I'm going to try and just give you a real brief overview, and don't go away confused, just go away excited that you can look at these techniques later and try and learn more about them, because these are the state of the art and this is where we're heading. So the first thing, which has been around for so long, is this protein chemistry or high performance liquid chromatography. It's been used for, since the 50s or even earlier to identify a whole range of different diseases and disorders. And basically, it's used to, this HPLC column here, it's used to separate, identify and quantify components in a complex mixture. So you can take a blood sample, for example, and put it through this column, and it can identify the various forms of the hemoglobins. And it can separate very accurately all of these particular hemoglobin forms. A, A2F, S, D Punjab, and G Philadelphia. The thing with this is it's very accurate, but it's time consuming. You have to have a nice, good, pure sample. Otherwise, you're going to get contamination. You have to have a really expensive piece of equipment to do it. But it's highly accurate. So big labs all over the world that have lots of money do this because of the fact that it's just so highly accurate. But as I said, it's quite expensive. But what happens is it separates out, so you put your sample in through this column, it's detected as it flows through this particular column, and the way it interacts with the medium in the column separates out the different sizes of the particular hemoglobins, and they come out. Sorry, Jane?
So is HPLC similar to electrophrasis? Is it on electrical charge or some other property of the hemoglobin?
It's based on electrical charge. So it basically separates out based on electrical charge, and you'll see something very similar in a second, and size. So what happens is that the gradient in the medium in the HPLC column will separate out certain charge molecules first, like the negatively charged or the positively charged first, and then they'll fall out based on this particular pattern here. So you see a whole range of hemoglobins that you can actually, globin genes that you can detect with HPLC. So it's kind of like the gold standard for testing protein molecules in complex mixtures, and as I said, it's been used for a long time, but it's expensive. The other method is IEF, or isoelectric focusing. Isoelectric focusing is a little different. This is based on the isoelectric point. So your proteins will migrate along a pH gradient, and then once they reach their isoelectric point, they'll stop. So you guys remember your isoelectric point chemistry stuff? So you can actually just take a blood sample. So what normally happens with this kind of work is you can take just a pinprick of blood and put it onto a card and send it in, and they punch out a small bit of that particular blood sample, put it into a tube and run it onto a gel here. And I don't know if you can see this really well, but you have low pH here and high pH here. And you have a whole range of you have your adult hemoglobin will migrate to a certain level. You have hemoglobin fetal, which migrates to here. And if you're testing for sickle cell trait or sickle cell anemia, they'll migrate at different levels too, based on their isoelectric point. And you can see here in the normal, we have hemoglobin A and hemoglobin F migrating to a certain point, isoelectric point on the gel. If you look at the sickle cell trait, for example, we have the sickle cell trait migrating here, and then this person over here is homozygous for sickle cell anemia because they don't have the A form. They have the fetal form and the sickle form as well. So isoelectric focusing is based on really identifying where the isoelectric point is of protein, and you know all proteins have isoelectric points, and it's all based on pH. Do you remember your metabolic biochemistry? Yep. So it separates proteins in a gel medium based on this pH gradient. It's very rapid. It's relatively inexpensive because it's a gel-based system. You can just buy the gels already set up and just put them on and run them. But there are some resolution issues. As anything, as you look at agarose gels, the cellulose acetate gel is different. That has really good resolution. If you look at agarose gels and running DNA, depending on the percentage, depending on the sample, you can get really poor resolution. And if you get poor resolution, you get fuzzy bands, and that can lead to a lot of false positives. So that's one of the disadvantages. Another one to look at is this capillary electrophoresis. And the way this works is that charged molecules are separated based on their electrophoretic mobility. So you place your samples in here, and they move along to this detection system here. So what happens is they move along this capillary and they elute based on their charge. If they're positively charged, they elute faster. If they're negatively charged, they elute last. So what happens is, because this matrix is all positively charged, if you have a positively charged molecule, it's being repelled along. And if you have a protein that has a lot of positive charges on it, it's going to be repelled even faster because there's more positive charge, okay? So so highly charged protein ions move faster than the less positive charged ones, and then the negative ions will also come out at the end. So you'll get a whole range of different profiles based on where they are on their charge. And those charges are well established, and you can actually look them up to find out, there's cheat sheets on what they are. And you get this kind of thing here, where you get these various peaks. And again, don't pay too much attention. It's hard to see these, but you can see various peaks as they come out. 1, 2, 3, 4, 5. As they come out of the column, you get the various species of globins, or whether they're hemoglobin G, Philadelphia, and a whole range of different things. It's time consuming. You have to have a really nice, accurate, really good sample. It's expensive as well. Again, you need that really elaborate machinery or equipment, but it's really accurate. It's highly accurate. Yeah, hi.
The capillary electrophoresis, they move from negative to positive, or from positive to negative?
That's a good question. It depends. It depends on the pH of the system. Because proteins are either negatively charged or positively charged based on the pH of the system. So if you want proteins to migrate from a negative to a positive terminal, you need to add a buffer system so the majority of the proteins are negative. So they'll be pushed and migrate to the positive terminal. So it all depends on the buffer medium. Do you remember that from your chemistry? Yep. Okay, and I know Jane talked about this one here, where we're looking at gel electrophoresis now, so I'm going to give you 2 examples that you guys are doing in the prac classes. But really, there's fast and slow hemoglobins, as Jane alluded to earlier. We have the fast hemoglobins, which migrate faster in a gel medium. Very similar, remember we talked about how DNA migrates in a medium? DNA migrates in a medium based on size, right? The larger the DNA molecule, the slower it migrates. The smaller, it can move through those pores very quickly and migrate towards the fastest. This is a bit different. This is based on the migration of their charge and how they interact with the actual the medium that they're in. And you can see that they're faster. There's faster ones and there's slower ones. And it's quick and inexpensive. You can just set up a gel, no problems at all. Again, there's issues with resolution, as I said just earlier, and I'll just show you a couple of those photos now. This is one of the methods that you're doing, is that right, Jane? Cellulose acetate? Okay, so cellulose acetate gels set at pH 4. These are really the primary screen that is being used. These are inexpensive, very easy to do primary method to detect abnormal hemoglobins. And the thing with this cellular acetate is that they have really quite good sharp resolution. So you don't tend to get a lot of false positives with this. It's really nice and sharp and clean. Good resolution. They can be a bit more expensive. I know you guys probably, are you making your own or you're buying them? So you buy them, they can be expensive. More expensive than just agarose gels. Agarose gels are really cheap. But they do give you really nice results. And as you can see here, they migrate to different levels. So if you look here, we have a control lysate, which contains all of the different this here is the well. Sorry, you can't see that. And this is different forms, A, S, there's a fetal form, and the A form here. That's strange you can't see that on there, isn't it? Yeah, it needs to go up a bit. And so they migrate at different levels, and here you've got hereditary persistence of fetal hemoglobin here, for example. So that's the F hemoglobin. So you can identify a whole range of different hemoglobins based on how they migrate in a gel, and it's quite accurate, quite sharp. If you look at agarose gels, they're different. Again, similar concept, but they don't have the resolution that the others do. Agarose gels are poorer resolution, they're inexpensive, but they don't give you the same kind of results. But they're quick and easy to set up. And again, you can see here hemoglobin A through here and a whole range of different patients and the various hemoglobins that have migrated through these particular gels. So is that the kind of stuff you're doing this week as well? So you'll get a sample, some samples, blood samples, and you'll be putting them on the gel and migrating, and you'll be able to know exactly based on a kind of a cheat sheet. You have a cheat sheet of where they should migrate to, which is well known. Hit my funny bone. It's not that funny. Oh, hang on a minute. It's just yeah. So they'll migrate all to different levels. Okay. I've got pins and needles. To try my left here. Oh, okay. So now let's move off that kind of macro level of molecular biology and move to a more micro level and DNA sequencing. DNA sequencing is kind of gold standard of identifying mutations because you can take a piece of DNA and if there's of a known gene, you can PCR the beta globin gene from a normal patient versus a diseased patient, and you can sequence it and find out. Oh, sorry, Jim. You can sequence it and find out where the mutation is, and then you can actually identify the particular disease state. Or if it's a new mutation that hasn't been found before, you can then translate that out into protein in silico in the computer to see if it changes amino acid structure or whether or not that mutation will be deleterious to the cell. So you remember what we did last semester, guys, when we looked at translation, and so now how important that is to understand that sort of stuff. This is where this comes in. So DNA sequencing, it's really quite easy to do. It can be sequenced and taken advantage of prenatal diagnostic cases as well. So you can get blood from the umbilical cord. So it's non-invasive. It's time consuming. I've done lots of DNA sequencing before and it's really time consuming. You have to have a really pure sample. You have to then make sure it's purified really well. You've got the right amount of quantity or concentration of it. Then you send it off, unless you've got a sequencing machine, which we used to have in a lab. But we got rid of it because it was just, they're like hundreds of thousands of dollars. We send them off now to AGRF, which is Australian Genome Facility, Research Facility, and they do all of the sequencing all over Australia, basically. And they sequence your DNA for you. It's inexpensive, it doesn't cost much. One sample costs you about $8. So it's not really expensive. And it's really highly accurate as well, as long as you've got pure DNA samples. And you get these kind of runs that look like this. So you can imagine if you had sickle cell anemia where you've got this mutation here, normal glutamic acid changed here for a mutant valine, where you get a change to a GTG from a GAG, you can actually sequence it and you'll be able to see that. And each letter, each G, A, T or C has a different dye attached to it. So it gives a different colour. So you can see clearly here G is black and then you've got an A which is green, a red which is T and so on. So you get this whole run. So you can imagine how easily you could find whether there was a mutation in a particular DNA sequence using this technique. Now, also another option is, I might just skip over this one because it's a little bit complex and I think you'll be more confused than Anna, because if you haven't actually looked at primer design and how they work properly, you probably will have trouble with this one. So do you mind if I skip this slide? But this is one that's important. A lot of diagnostics in disease states, cystic fibrosis, these particular sickle cell anemia can be identified through what's called restriction fragment length polymorphisms. So do you remember what restriction enzymes are, guys? Bacteria produce those enzymes. They're like the bacteria's immune system. When a foreign piece of DNA, a foreign virus, starts to invade a bacterial system, the bacteria says, hey, what's going on? And secretes a specific restriction enzyme. This restriction enzyme is like a scissors, then it goes along and it cuts up all of that foreign DNA material. It doesn't cut up its own DNA, which is pretty amazing because its own DNA is methylated at those sites where the restriction enzyme cuts. Restriction enzymes are very specific for certain DNA sequences. They'll only cut at certain sequences. So what happens is the bacteria will secrete these, they'll cut at certain sequences in a viral piece of nucleic acid, cut it up, render it useless. It won't touch its own DNA because at that specific site where they normally cut, it's methylated, it's protected. So it's pretty amazing that evolutionary, think about in evolutionary terms how this has been designed. But we use restriction enzymes in the lab to cut DNA because we know there's a whole range of flavours of them. E. coli produces ECOR1, which is a restriction enzyme that cuts at GGTTAA. That's where it cuts. It doesn't cut anywhere else. So there's a whole range of different flavours. But we can use those in the lab. There's this MST2. MST is a restriction enzyme site in DNA. And in the normal globin gene here versus the mutant globin gene, you have the normal globin gene here. This MST cuts at two sites and creates two bands. It cuts at two sites because there's two of those DNA-specific sites in that gene. But the mutation that occurs in sickle cell here abolishes that cut site. It's gone. So it's a really nice technique to use because now the mutant beta sickle cell globin gene only has one site that it cuts at, not two. So a pretty easy diagnostic test then. So you can take a sample of normal and a sample from someone that you suspect might have sickle cell, and you can add restriction enzyme to it in a tube, put it at 37 degrees for an hour, run it on a gel, and you'll get some sort of pattern that looks like this on a gel. And you'll see here, for example, a number of different things here. So you've got this father here is a carrier, so they'll contain the two cut sites here as per normal on one allele, but the other allele, they've got a single band, which is this one here. So they're a carrier, okay? Jane?
Do you use this sort of technique for population screening?
Yes, you can. This is a really good question. There's large-scale genome sequencing of populations that are susceptible to various diseases in specific geographical locations, different populations, different dietary requirements, a whole range of things. And you can actually screen using this particular method. One of the Han Dynasty from China, which is I think in the 12th, 13th century, their relatives today all have a mutation. They have a high susceptible rate to type 2 diabetes through this mutation in a particular transporter. And it's been passed down through generations and generations. So you can screen a whole population of people from that particular heritage and this way and find out, as Jane said, in a large population studies, whether they're susceptible to getting a particular disease or not. So, and then the mother here is a carrier, so she has one allele, two cuts, and then here. What's this circle mean again? Boy or girl? Girl, right? Yep, good. So it's not coloured, what does that mean? No, what? Sorry? No disease, perfect. So would you expect to see a band up here? How come? You wouldn't, you think? No, how come? Sorry? Perfect. Beautiful. So both alleles have the same cut, right? But there's two alleles, remember? In this one here, one allele cuts twice, the other allele cuts up here, so you have this carrier. But here, both alleles have cut here, and they just sit on top of each other because they're the same size. If you actually ran this on a gel, these two bands here should be darker because you've got two lots of alleles sitting on top of each other of exactly the same size, right? Now, what's this square mean? Men. What does the colour mean? Disease. Okay, so would you expect anything in here? No. So are they heterozygous or homozygous for the disease? Homozygous. Perfect. So they carry disease, and then the girl here is a carrier. So this is a really nice way to test families, and you can do this in subsequent generations. Remember we talked. Sorry, guys, if I'm talking stuff that you haven't heard because you're not in the class, but we talked in human molecular biology the other day about the different generations that occur in these pedigree maps. We can actually continue in the third, fourth, fifth, sixth generation, and we can actually figure out the frequency or the percentage of potential people that will be carriers or have be homozygous for the disease. Now, there's another method called real-time PCR. And you guys are going to be doing this in your prac class. I don't want to go into too much detail about this because it does tend to get really quite complex. But the idea is to design probes with different melting temperatures. So the probe with the lowest melting temperature, or the TM, lives over the mutation. So you design these probes that sit, probes are nucleic acids that sit on the mutation, and you design them in a way that they sit over the mutation. And what happens is that when you're performing PCR, the TM is and if you look here, the TM is this melting temperature when half of the species of DNA in your sample has been denatured and melted. So if you have a probe that has a lower melting temperature than another probe, and that probe has a fluorescent signal on it, you can detect how they melt and at what degree they melt. And then you can chart them to find out if this particular probe for mutation A versus this particular probe for mutation B melts differently, you will see that. And then you'll know what the mutation is. A single base mutation can shift the melting temperature by 5 to 10 degrees. So the whole idea is to look at real-time PCR is to design these probes across various mutations. And you see here, this is the beta globin F, and what you have here is a number of whole different mutations, T, G, C, and so on. So these probes are designed over those sites, and they're designed in a way that they'll have a different melting temperature. So you can throw all of those three probes into the same DNA sample, do the PCR, and then what comes out will give you a test on whether they have this mutation, this mutation, this mutation, or all three, or two, and so on. Quantitative real-time PCR we'll do in our prac classes. It's very expensive, but it's extremely accurate, and it monitors things in real time. So you get to see them happening in real time. You don't run agarose gels. The way it works, and I'm just going to go into this really briefly, is that real-time PCR works where you design a probe that has a specific dye attached to the end of it. And that specific dye is a normal probe which will bind to the normal DNA template. So it will bind here, the G to the C. That's a normal DNA template. The blue is a mutation in a particular DNA template. So the T here will bind to the A. So where you should have a G and a C, you have a T and an A. So you can imagine you could detect sickle cell anemia this way. But it has a different probe on the end, a blue probe. What happens is when you do the PCR, the primers move along the DNA and they hit the probe and they just dissociate the probe. They just release it. They bash into it and the probe is released. And that probe that fluorescent probe gives us fluorescence that then we can measure. So for every probe that's released, it's one fluorescent unit. So we can measure fluorescent units this way. And if you have the mutation, you'll release the blue probe and not the green one, and so on. So you get something that looks like this. And again, don't worry too much about it, but what you'll see is, and I didn't show the red probe is normal. So the normal axis is normal allele on the x-axis. The y-axis is mutant alleles. And you'll see green are carriers because they cross from the y to the x-axis, so they're carriers. And you have to know the genetics behind the probes, how they're designed. But don't worry about this now. We'll go into that in detail later for you guys. And, the blue, because it just sits up here on the y-axis and doesn't transverse across the x-axis, these are homozygous. They have the two alleles that are mutated. Okay? And again, understanding this is understanding how these probes are designed. I don't want you to understand that now. I just want you to have an idea that we can detect from a population a whole range of different genotypes, I should say. Okay, another thing that I want to talk about is digital PCR. This is really the state-of-the-art PCR at the moment. So digital PCR is that you have this kind of chip with 20,000 wells in it, 20,000. And you load your sample in and it mixes with oil and these droplets. So every single oil droplet, there's 20,000 of these guys, has its own PCR reaction in the oil droplet. It has one piece of DNA and it has all of the bits that you need for a PCR in one single droplet. And you've got 20,000 of them. So the beauty with this is, if you've got a really rare genetic disease, and you're mixing it with, you're looking for it in a DNA sample, it's going to be swamped out by the wild type sequence in normal PCR. You won't detect it because there's too much competition for the reagents to pull everything to the wild type because there's so much more of that. So much more of the normal sequence. So it just basically pulls out or dilutes out. Or what's the word in chemistry? What do you use? What's that word in chemistry you use? I use it all the time. I can't think of it now. I can't think of it. Anyway, so basically you pull out all the reagents in the reaction towards the wild type and not the mutant form. So this method here puts the mutant form in its own little compartment. So there's no competition. It carries out the PCR in its own little bubble. So this is the beauty of it, and I'm not going to go into too much detail, but the PCR is performed, and then all you just do is you count the dots. You count how many, it's a fluorescent probe-based system, you count how many light up. If you design a probe that's a specific colour to measure a specific genetic disease, it comes up with that colour. So for example here, if you designed it to be green, and grey is normal, you can count how many particular fluorescent molecules light up gives you an indication of the disease phenotype or the severity of the disease. So if you've got something like this, high concentration, you've obviously got a high number or a high copy number of that particular mutation. If you've got just a few like this, you've got a low copy number of that particular mutation. Okay. And we can use this to detect various copy number variants in thalassemia, for example. And I won't go into this in detail, but copy number variants are, you have the specific gene and it's copied many times, and there's a whole range of copy number variants. So you can use that particular digital PCR